I fervently hope that MTE is not just about competing and winning awards. Most importantly, it has to be about cultivating an innovative mindset within the society, bringing entrepreneurs as well as scientific and technology tech communities together to find common ground by collaborating and working towards bringing better technology solutions, innovative products to the world.
I hope that this 3-day event will provide substantial opportunities for participants from multifarious backgrounds to not only display their innovations and inventions, but also to seize the opportunity to interact, discuss, and exchange ideas on the latest techniques and technology that will eventually benefit all, inshallah.
MTE provides a conducive platform for innovators to promote their invention through various innovative approaches, as well as to expand. So this exhibition also allows them to find potential investors or customers to help in marketing and commercializing their invention products in the market.
We need to continuously inspire, ignite and innovate our youth to make sure that they will become prosumer.
We want to thank our partners, Proton Group and team, with over 20 years experience in organizing such an event for making this possible.
We came from Poland,
Vietnam,
country Croatia,
Iran,
Taiwan,
Bali, Indonesia.
So far it's been extremely interesting to see a broad range of technical and scientific endeavors.
We made a lot of contacts regarding next step with university community and with real sector community.
We are very glad we are here and for this great exhibition.
Very high performance, and that we show what the technology we have.
It is indeed my privilege to welcome all participants to the Malaysia Technology Expo 2020 Special Edition the COVID-19 International Innovation Awards. Thank you for joining us, especially all the international participants.
Saya bagi pihak NADMA Malaysia mengucapkan terima kasih
syabas kepada MTE 2020 COVID-19.
It is my pleasure to join you today at the Malaysia Technology Expo MTE 2020 Special Edition COVID-19 International Innovation Awards ceremony to recognize and celebrate the hard work of those among us who constantly push the boundaries of innovation. I would like to congratulate all the winners as well as your team members who have been supporting you throughout your journey.
MTE 2021 Virtual Edition will be the catalyst in encouraging innovators and inventors to share and inculcate innovativeness and sustainability in their research. Innovation is not just about making new and better products. It encompasses all aspects of ideas, processes, systems, services, methods and models.
As large gatherings are no longer encouraged, I'm happy to see that MTE has gone virtual. This will allow more innovators and to promote their innovation and share their ideas with the world.
We look forward to university putting a strong efforts to forge strategic collaboration with industry to generate new sizes, innovations and commercial entities.
There may come a day in the near future when we have overcome this crisis, but our innovations may be able to assist another country in their fight against the pandemic.
To lift student engagement and attainment in innovation and to support teachers and improve student outcome, innovation and technopreneur mind.
Involving youth at an earlier stage is important, especially as Malaysia is slowly but surely becoming a greying nation. Innovation alone is not sufficient. We must also create an infrastructure for incubation of ideas and transform them into successful ventures.
Enhancing not only in the design but also the delivery of public goods and services for the people. Innovation in government is imperative not only to transform but also to stay ahead of change.
The quick responses to protect the citizens from COVID-19 shows the unity of the nation. Know that your contribution is critical to the transformation of our country.
I'm fully confident that this year's MTE IIA, albeit on a new normal platform and approach, has achieved its desired impacts.
It has been nurturing the innovation culture among scientists, researchers, students and startup founders in order for them to transform their ideas into business ventures.
Ladies and gentlemen, our next lined up program is a knowledge sharing session. Please enjoy.
Assalamualaikum and good afternoon, everyone. Thank you for inviting me to be the speaker for today's webinar and for giving me the opportunity to talk about how civil science could ease testing pressure and track COVID-19. In today's webinar, I will give a brief introduction on what is COVID-19 virus, what do we currently know about sewage and COVID-19, how sewage testing complement current clinical testing, what has been done so far by the sewer scientists on wastewater testing to tackle COVID-19, as well as how sewer scientists perform such testing. Now, this map here is taken from European Center for Disease Prevention and Control. It shows the geographic distribution of COVID-19 cases worldwide as of yesterday. So the red spots on the map indicates the COVID-19 cases. The bigger the spots, the more people are infected at that particular country. So as you can see from the map, COVID-19 has been spreading rapidly throughout the globe, resulting in more than 10 million confirmed infections as of this morning and about 500,000 deaths, which has presents the world with huge challenges. So COVID-19 is caused by severe acute respiratory syndrome coronavirus 2, also known as SARS-CoV-2, or we do call them as COVID-19 virus. So it causes respiratory illness. The major transmission route of this virus is known to occur via hand to face, from nasal pharyngeal to eyes, nose, and mouth, contact with contaminated surfaces, and inhalation of aqueous aerosols, including cough and speak, including when you cough and speak. So this is why our Ministry of Health always remind us to avoid 3C, which is crowded place, confined space, and close conversation, and also to practice 3W, which is wash hands frequently, wear mask, and warn. So this is also a new strain of coronavirus that had previously not been identified in humans. COVID-19 virus is distinct from SARS-CoV that emerged sometimes around 2002. This also means that even if you have been exposed to SARS-CoV previously, you will still get the novel coronavirus. And it is also a new zoonotic virus. We call it as zoonotic virus as it jumps from animals to human. So how this virus gets transferred to human? Now, imagine that this virus underwent a mutation within the bat, and then it mutated enough that it gets transferred into another animal as an intermediate host. However, to date, scientists, we are not completely sure about, but we believe that it could be pangolins mutated enough and can now infect the human and cause COVID-19, which is the novel coronavirus discovered last year. So why knowing the intermediate host is important? This is because pangolin, according to some research, its scales has some healing properties and is used in traditional Chinese medicine to help with lactation difficulties and arthritis. Therefore, it is highly trafficked all around the world, which makes this very dangerous and increase the risk of pandemic. So the COVID-19 virus is highly pathogenic, meaning it causes illnesses. This virus also is virulent, which means that it has harmful effect, and worse still, it spread very quickly. It originated from Wuhan City, China in December last year and was declared a global pandemic on the basis of alarming levels of spread, severity, and inaction by the WHO on March 11 this year. So on 25 January 2020 this year, the first COVID-19 was detected in Malaysia, and traced back to 3 Chinese nationals who previously had close contact with an infected person in Singapore. Because this is a new virus, there is basically no immunity in the population, no drugs, and no vaccine available yet. So this map here shows us the total number of tests per 1,000 people compared across the countries. So as of yesterday, the US has performed over 32.6 million tests for COVID-19, which is the second highest number of any country worldwide. Last Monday, the COVID tracking project data shows that although US is able to perform 500,000 tests a day, every day, started from June 19 this year, but many states are still falling short. This shows that even a developed country like US, they are only able to cover less than 0.2% of their population a day. Therefore, it's more likely that some developing or least developed country will be falling short of testing capacity, especially if they have larger population at risk, fewer resources, and less capacity. This data clearly show that every country cannot cope with testing everyone, as it is not simple, easy, or cheap. Now, last month, researchers in the Netherlands has found signs that the COVID-19 causing virus can be detected in sewage. Soon after, several other countries around the world, such as Australia, the US, Spain, Italy, Japan, Indonesia, and Singapore, also started their own sewage analysis as a possible outbreak warning systems. All these findings clearly indicate that sewage surveillance could be a useful tool in detecting whether COVID-19 virus is present in the population and to help monitor the spread of COVID-19 while reducing the need for mass testing. And we even see headlines like, sewage could hold the key to stopping new coronavirus outbreak. Now, two interesting findings by Dutch scientists where they detected the presence of COVID-19 genes in the sewage water. In one of the studies, they detected COVID-19 genes at a wastewater treatment plant in Amersfoort on March 5th this year, before any cases has been reported in that city. So another study also done by Dutch scientists, what they found is that there is no COVID-19 virus detected in the wastewater treatment plant in Tilburg and Kosovo until the first week after the first confirmed cases occurred. So these findings are significant as it could pave the way to develop a monitoring system in the future against impending COVID-19 outbreak. Now, you must be wondering, why is there COVID-19 gene in the sewage system if this is a respiratory virus? So most people know that they will emit loads of this virus through respiratory particles in droplets from the lungs. But what less well known is that they will also emit small virus particles in feces, especially for those who experience diarrhea as a symptom. And these virus particles also shed in stools. This was supported by Zhang group, where they found COVID-19 gene in 5 of 14 fecal samples tested, as well as Cell group, where they found COVID-19 gene in 39 of 73 fecal samples from hospitalized patients. Now, positive isolation from feces persisted in 23% of the patient in this study, even after it disappeared from the respiratory tract. So some other clinical studies reported asymptomatic or pre-symptomatic individuals can also shed virus particles in their feces. This was supported by Jiang group and Ling group. Both reported that COVID-19 gene could be detected in the feces even with a negative swab result, which makes this the bonus for sewage monitoring. And what we not yet know for sure is whether COVID-19 virus particles are culturable or not. There are two papers that shows that they are able to culture these viruses from stool. Both were done in China. But a more recent paper from Germany was not able to reproduce the result despite high amount of COVID-19 gene, which might be due to the differences in protocols for virus isolation. So the importance of this is we are detecting the COVID-19 gene in wastewater sample. Therefore, whether the virus is still infectious is not our main concern. So this finding has raised the possibility of COVID-19 can transmit by fecal-oral transmission. While data are limited, there is no information to date that anyone has become sick with COVID-19 because of exposure to wastewater. However, we can't rule that out. Therefore, it is encouraged to protect wastewater workers by practicing hygiene precautions and PPE when handling untreated wastewater. So, what is wastewater-based epidemiology, also pronounced as WBE? So it is an important tool for monitoring the circulation and prevalence of COVID-19 through wastewater. It detects and measures chemical signatures in the sewage, such as in this case, the COVID-19 virus gene. And it is quick and simple to perform. So what you need to do is to identify wastewater treatment plant and detect the COVID-19 gene using molecular method. And the result allows you to assess disease prevalence and spread in a defined population. Now, although WBE cannot identify which individuals have been infected, but it has several advantages compared to clinical screening for each individual. Since WBE is a blind to biasness and test on the whole population that shared the same wastewater treatment plant, it provides a more comprehensive and objective data, and it shows the whole trends. It does also have a great potential as an early warning detection, where it is able to predict COVID-19 outbreaks 7 days before individual clinical testings and 3 days before hospital admissions. Another advantage is WBE allows the identification of hotspots in a localized region of population, as well as estimate the total number of infected people at a given time. It would then also be enabled to compare the infection rate between communities, facilitating the prioritizations of resources for control and mitigation plan. Moreover, WBE also allowed to assess the effectiveness of interventions such as lockdown, social distancing, and reopening steps of the economy. Basically, it provides a very effective tool to monitor the changes in the COVID-19 levels in the population due to various factors. So now, WBE is not a new thing, and it is a well-established method that has been done on global scales for other purposes such as monitoring drugs consumption, lifestyle choices, as well as monitor infectious diseases and the spread of epidemics at the community level. So the 1 liter of wastewater that we collected is like the fingerprint of the community. Whether we do, whatever we do, we eat, we consume, we use, we leave a fingerprint in wastewater. And using advanced analytical or molecular methods, we are able to detect the chemicals or pathogens. Now here are a few successful stories on WBE in monitoring outbreak. So polio is the most successful story of WBE. Environmental surveillance of polio in wastewater has been utilized since the 1980s, year before when the term WBE was coined with Finland, Israel, and Senegal. All successfully analyzing sewage samples in order to assess polio circulating within populations. WHO have also released guidelines for employing environmental sampling to monitor polio in wastewater sample. So wastewater surveillance have also demonstrated promising results with the potential for retrospective prediction of disease outbreak such as hepatitis A and norovirus in Sweden. And influenza in wastewater was also investigated during the H1N1 swine flu virus outbreak. So influenza A viruses were detected in sewage. WBE was also used to monitor lifestyle choices, varying from alcohol consumption, tobacco, and psychoactive substance. So this study here shows that the highest level of alcohol consumptions were collected in the metropoles and brothels compared to smaller villages by quantitating ethanol metabolites in wastewater. So this is the weekly pattern show that there is a clear week and weekend difference in alcohol use with intermediate levels on Monday to Friday. Well, this study studied the tobacco consumption in local communities in Italy, and they used the data to assess the efficacy and efficiency of tobacco control interventions with the aim to design and implement effective tobacco control plans. Now, WBE was also used to detect illicit drugs abuse and harmful chemical screening in the US and in many other countries. So this study here, this shows the seasonal changes in the concentrations of 21 different pharmaceuticals in a wastewater treatment plant. And they investigate over 12 months. So this process allowed back calculations of community-wide drugs consumptions or exposure to chemical factors and can provide unbiased reflection of key aspects of public health. Now, to fight against COVID-19 pandemic, monitoring is important. As again, moving forward, what monitoring can help us is that we cannot stop the transmission altogether, that it cannot be zero. But what we can do is that we can prevent the exponential increase and prevent the spread of the virus exponentially. WBE is another area that can help in proactively identifying and containing new outbreak by complement the increased clinical testing and enhanced contact tracing efforts within a population. So this map here shows what is happening in each state in Malaysia. Sorry that I am not able to find the very recent one. So as of June 29, COVID-19 virus has infected around 8,637 people and 121 confirmed deaths with 8,334 recoveries, according to our Ministry of Health. So the good news is that across Malaysia, the MCO since March 18 has successfully leveled the pandemic curve in the country. So Malaysia is currently seeing only a handful or less of new cases each day. As activity gradually resume in Malaysia during this RMCO, we know that there will still be a risk of new waves of infection, and this will last until a vaccine, treatment, or herd immunity is found. Therefore, knowing the actual outbreak scale is important to understand the effectiveness of mitigation strategy. So obviously, there is a clear need to properly monitor the spread of COVID-19 virus so that we can continue to flatten and lower the curve, especially in the near future as we might be hit by new waves. Also remember that not until a vaccine, drug, or herd immunity is found, there is really no cure to this. One of the essential methods used to monitor COVID-19 outbreak, where all countries have been doing it, is direct testing of the public. It is also called as clinical testing. However, we also need to be aware that direct testing of public has its own limitation. As I mentioned earlier, even countries that have the resources or capacity to test each individual, with most only able to test healthcare workers or people with symptoms severe enough. That also means that we will miss out people who are asymptomatic or pre-symptomatic. Bear in mind that there is a lot of people who have no typical symptoms but have the virus with them. Technically, these people will not go to the hospital, will not get themselves tested, and so it will bring some question about the statistic representation of data of the overall population and some bias related to that. For example, the number of confirmed cases reported daily is not able to reflect the actual scale of outbreak, since those numbers is only based on confirmed cases and would not reflect mild and undetected ones. Also, the number of confirmed cases are solely based on the number of samples tested each day. This also means that authorities have only limited information about how widespread the new coronavirus is, or whether it is affecting some communities more than others. Now, other limitations such as limited coverage. End of last month, our Director General of Health, Dr. Noor Hisham Abdullah, announced that Malaysia's lab capacity for COVID-19 testing will reach up to 30,000 per day by now. If we take this testing figure into consideration, it will take nearly 3 years just to test the entire population, with a sampling rate of less than 0.1%. Well, other countries are also facing the same problem. Another option to monitor COVID-19 is through contact tracing. This is one of the core strategies to contain the spread of COVID-19, which is what we are currently applying in Malaysia. For contact tracing, we do have the technology to it. Our government has also launched few smartphone apps such as MySejahtera, MyTrace, Gerak Malaysia, and few others to help health officers track the spread of the virus. However, not everyone is very cooperative in providing their personal information. Moreover, contact tracing is also very resource intensive. Although the technology part is pretty easy, but you basically need the army of trained people to do the actual contact tracing. Over here is the recent article published in US, where the Johns Hopkins Centers for Health Security says that more than 100,000 trained tracers are needed to perform contact tracing, and obviously it is quite difficult to achieve that. So another option to monitor COVID-19 outbreak is through WBE strategy that I will be explaining more in today's webinar. So we need to know that there is no silver bullet here. WBE also have its advantages and limitations as well. So this table shows the amount of time needed for different testing methods. It takes approximately 3 years to test every single individual in Malaysia with the current testing capacity per day. Even if we test all the wastewater treatment plant in Malaysia, it will take approximately 1 day to do so. So this also means that WBE strategy can be used to monitor the entire Malaysian population and track the spread of coronavirus. Moreover, this strategy could serve to better target and direct the application of clinical testing. Thus far, Sewage scientists has used 3 basic forms of WBE to monitor the spread of COVID-19 in the community, namely the qualitative, that only indicates whether COVID-19 gene is present or absent, quantitative, that capable of indicating the amount of COVID-19 gene, and lastly, estimating the infected population, where it is able to estimate the total number of infected people in a given time. Specifically for COVID-19 application, how could WBE be used? So one of the first study published on this was from the Netherlands, and what they have shown is they measure the COVID-19 gene in 7 sewage treatment plants, which is the orange line here, over a period of time. And they also plot the number of COVID-19 hospitalized, which is this gray line here. They noticed that there is a very good correlation between the number of cases and the quantity of COVID-19 gene that are measured in the wastewater. Another example is from France, and so this light gray is the number of consultation, dark gray is the hospital patient, and they estimated the concentration of COVID-19 gene and also have a very good correlation with the number of cases in France. This study is the first study that reports the detection of COVID-19 gene in wastewater in Australia. So what they do is they use the estimated viral RNA copy number observed in the wastewater to estimate the number of infected individuals in the catchment via computational simulation. The model estimated a median range of 171 to 1,090 infected persons in the catchment, so which is in reasonable agreement with clinical observations. Now, this study is from US, has been published very recently. So what they have done is instead of measure the virus in the wastewater, they measure the virus in premises. So they take from untreated sludge. So, and what they seen is that in the very beginning of the outbreak, the concentration of the virus in wastewater is in fact much more sensitive compared to the number of patients data. So here is the figure. Here, this figure clearly shows in the red line, which is the concentration of COVID-19 gene, and the black line is the number of cases, and there is approximately 1 week delay. You can see this 1 week delay in detecting the first positive cases. This is extremely critical because as soon as the patient becomes infected, they started shedding the virus. So we can detect the virus earlier in wastewater, but having the clinical test and then getting the result, it does take some time. So there is a delay that the patient will need to develop the symptoms first, then they feel sick, then only they go and see the doctor and get themselves tested. So this time delay here in such outbreak is very critical, and 1 week can be very helpful in preventing the uncontrolled spread of the virus. So this is the importance of wastewater monitoring for COVID-19. Another similar study from Spain. This blue line here is the concentration of COVID-19 gene, whereby the red line is the number of declared active cases. And if you can look at the date over here, there is approximately 2 weeks delay in detecting the first positive cases. Again, it has a good correlation with the number of cases. So nearer to our home country, Singapore, early this month, they had also started extracting wastewater from men's hole at migrant worker dorm to test for COVID-19 gene. The trial result shows that the amount of COVID-19 gene in wastewater is related to the prevalence of COVID-19 in these living spaces. Findings for dormitories in this pilot program with no detected COVID-19 case and no COVID-19 genes in the wastewater will provide added assurance that the dormitories remain free from infection, and so they are allowed to leave the dormitories for work. Now I'll stop here for a short break. After the break, I'll talk about WBE workflow.
On behalf of the Water Loss Industry Group, I would like to congratulate the Malaysia Technology Expo on its 20th anniversary, wishing you many more successful years.
This is Srijana Gajmer from Bhutan, and I'd like to wish MTE
on its 20 years of sharing, learning, and growth. I wish them many more glorious years of the same. Thank you.
Okay, now let's continue. So before going into the details of the method, let's take a look at this video so that you get an idea on how small this virus is. So imagine these tiny viruses floating in the 1 liter sewage water, 1 liter of the sewage water. So along with many other particles that are much larger, this poses extra challenges in the processing of sewage samples and extra steps involved to collect the virus and remove the contaminants as opposed to swab testing. So this is the technical workflow for COVID-19 gene detection in wastewater. Basically, it involves wastewater collection, virus concentration, RNA extraction, RNA amplification, and measure of the status of coronavirus infection at population level. So I'll go into detail of each step. So detection of virus material in wastewater begins by collecting wastewater sample. So two different methods are being used to collect sample: composite or grab sample. For 24 hours composite samples, and automated equipment will be used to collect the samples at regular intervals during a 24 hours period, or during a peak hours of household sewage flow. Or it can also be done manually at predetermined time to form a time-adjusted composite sample. So here are some pictures showing how 24 hours composite samples is being collected. Whereas in the grab sample method, an amount of raw sewage is collected at a selected sampling site at one time. So once we had collected wastewater samples, we will then need to concentrate the virus particle in wastewater. Since virus particles in wastewater are very much diluted, therefore, different methods have been applied to concentrate virus in wastewater, such as filtration through 0.45 micron electronegative filter membrane. This uses the electrostatic interaction between filters and viruses to concentrate the virus. Okay, thank you. Okay, so other method Other methods include polyethylene glycol precipitation, also known as PEG, or PEG-coupled aluminium precipitation, aluminium-driven flocculations, ultracentrifugations, and centrifugal ultrafiltration, which is based on size exclusions, have also been used for concentrating viruses from wastewater samples. So, extraction methods is either manual or automated, are being used to remove impurities and purified viral RNA from the concentrated viral material. You can either use commercially available viral RNA extraction kit or conventional method for extraction. So extraction usually involves 5 steps. So first we break the virus by using specific buffer. And then you add ethanol to precipitate the RNA. Let the RNA bind to the column, perform few washes, and then elute the RNA. Finally, the RNA is reverse transcribed to cDNA and amplified via quantitative reverse transcription, PCR, which also known as RT-qPCR. So this amplification plot shows the accumulation of fluorescence signals with each successful cycle of amplification. So if you see the lower CT value, it indicates more virus is being detected. If we are performing qualitative analysis, we will then stop at this stage. But if we wish to obtain quantitative results, we will then need to plot standard curve by making serial dilution of the COVID-19 gene with known concentration. We will then calculate the viral copy number in the wastewater from the standard curve generated. Thus far, the currently available test assay for COVID-19 virus mainly targeting at ORF1AB region that code for RNA-dependent RNA polymerase. Normally people will call them as RDRP, which is believed to involve in viral replication. Another target is E gene, which code for the envelope, which is responsible for morphogenesis, and also N gene, which code for nucleocapsid that provide nuclear import signal. It also responsible for interfering cell processes, virus replication, and RNA package. So this image here is the cross-sectional of COVID-19 virus with its major element including the spike protein, viral envelope, helical RNA, and protein and membrane. So once we obtain the number of viral copy number in the wastewater, we will then estimate the total number of infections in a community. This formula here is used by Ahmed and his colleague to calculate the number of infected person. There are different ways to calculate infected population, but they are more or less the same. So RNA copies per liter of wastewater is the amount you obtain from RT-qPCR analysis. This liter of wastewater per day is from the Sewage Treatment Agency. How much a person defecate each day? This information, you can get it from previous publication. And viral load in the feces of COVID-19 patients. This also is based on publication. This value here is based on the publication here. So it can range up to a few hundreds to millions. So this is one of the limitations of WBE strategy, as the amount of viral particles shed in feces vary hugely between individuals. So now, what can we do with all these data that you obtain from WBE analysis? So if it is a qualitative analysis, you can use it as an early warning tool to identify the hotspots for more targeted clinical screening and enhanced contact tracing efforts within the population. It may potentially identify undiagnosed population at the population level as well. So we all know that we just entered the 23rd day of our MCO, where almost all economic sectors and additional freedoms for travel, sporting, and social activities have already opened up. So far, we are still able to continue flattening the COVID-19 curve, thanks to our government, especially our Health Ministry and Home Affairs Ministry for their hard work. Although we are now feeling more relief, but this is not the time to be self-satisfied. It is everyone's responsibility to maintain the MOH guideline and prevent new wave of COVID-19, which has already occurred in some other countries like US, South Korea, and China. So I obtained this graph here from Florida Department of Health, a very recent study from US published in the middle of this month. So, and what you can see here from the graph is, as soon as the second phase of economy started to open, there is a spike again in the number of cases. This one here is from South Korea, second wave of COVID-19 that involved at least 9 nightclubs in the nightlife district of Seoul. These new clusters was believed to emerge when a 29-year-old resident of Yongin City tested positive on May 6 after visiting these clubs in Seoul. So, how can WBE strategy play a role in tracking a second wave of COVID-19? It is by monitoring wastewater for COVID-19 gene at a regular interval. Once there is an increase of viral load in the wastewater, we will then know someone in that particular community is having COVID-19. We will alert the local health authority to begin mass screening for that particular community. Let's say if you wish not to stop at qualitative analysis, but wish to continue with routine quantitative WBE analysis, it will then allow you to illustrate the timing and scale of outbreak, where you can monitor the trend of both clinical tested positive patients and the concentration of COVID-19 gene. It also allows you to assess the effectiveness of the mitigation plan. Other than using wastewater as a surveillance system to control the spread of COVID-19 and effectively managing future outbreaks, if let's say genetic material of COVID-19 is detected in the wastewater, you can then move on to sequence the virus genome. This information can then be used to track mutations since viruses tend to mutate as they spread. It can also be used to study what virus strain that get transferred to the community, as well as using those information for drug and vaccine development. So this is the NextStrain project to track how fast COVID-19 virus travel across the globe. The project uses genome sequences of virus derived from patient samples to generate its map to visualize how this mutation changes over time. The circle here indicates the size of the outbreak from last December to this April in a particular country or region, and the color line indicates where the virus traveled. Let's see how fast this virus travels. was done in our home country. It is a cumulative cases in Malaysia until 31st of this year. Six of them are from first wave and 4 from second wave. So these 2 waves of infections were separated by 11 days, during which no cases were reported. Case of the first wave was associated mainly with travel from China and Singapore, while second wave were associated with religious gathering. This strain here was linked to a religious mass gathering, which has been associated with 48% of national cases and clustered with strains from Japan, Australia, and Saudi Arabia. Whereby this strain here is the cluster of the previous strain, but this patient from whom it was isolated has no clear link to the gathering. So this suggests that the strain associated with the strain associated with the gathering have established community transmission. Whereby this strain here from a patient with no history of traveling or attending gatherings, suggesting local evolution. So in conclusion, not until a vaccine is found, COVID-19 is expected to come back in waves. Hence, it is important to monitor the spread of COVID-19 virus closely. Although an accurate predictions of the infection population by WBE may not be possible due to several limitations, but this method can serve as an important tool to trace the circulation of COVID-19 virus around Malaysia. It does also have a great potential as an early warning system to identify areas for more targeted testing, as well as an indicator to track the scale of pandemic in the country. Therefore, testing wastewater can complement clinical testing by providing a quick and cost-effective way of tracking the extent of infection at population level, and also capture asymptomatic or pre-symptomatic individuals. Before I end my presentation today, I would like to share this tweet by Rika Davis. What he tweeted here is wearing masks could practically block all of the bacteria. Although this is not COVID-19 virus, but it did show us how a surgical mask blocks most of the droplets. Wear mask if possible, and as that is one of the best we can do for ourselves apart from washing hands and practice social distancing. Thank you for your time. So this QR code over here is a collection of posters that we used during our environmental COVID-19 testing campaign. So do take some time to scan and have a look at it. Let us know if you need more information. You can always reach us through this email and contact info. Thank you.